Thank you very much indeed, Clive. I'd like to welcome you all by ringing our bell, which formally starts our meeting. I would like to start our meeting where our last week's meeting finished. Before we get into any other business, I'd like to invite past President Hugh to tell us about the Saints' Day that we celebrated last Monday in absentia. Hugh, would you care to say a few words about St. Andrew?
Sure. Thank you, David. Thank you. Everyone associates St. Andrews with Scotland, but actually, St. Andrews is also celebrated in other countries, including Cyprus, Romania, Barbados, Greece, and Malta. And they all celebrate it in their own way. According to Christian teachings, St. Andrew and his brother Simon Peter were fishermen in Galilee when they became the first disciples of Jesus. Like Jesus, Andrew was martyred for his beliefs and chose an X-shaped cross on which he was nailed on 30th November 60 AD in Greece. The diagonal cross was adopted as his symbol and became the white cross on the blue flag of Scotland, known as the Saltire. 30th November was the date of his martyrdom, was thus designated St. Andrew's Day. Apparently, St. Regulus, a Greek bishop, was instructed by an angel to take some of St. Andrew's relics by ship and sail in a westerly direction, and where they landed or were shipwrecked, to erect a church. They were shipwrecked at a coastal village in Scotland called Kilrimont, and this later became known as St. Andrews, now famous for its university and golf. Sadly, the relics in the cathedral were destroyed in the Reformation, but the Archbishop of Amalfi gifted a piece of St. Andrew's shoulder blade so that the saint could remain forever in Scotland. Although he never set foot in Scotland during his lifetime, since the year 1000 AD, the Scots had been celebrating his honor, and he was declared as the nation's official patron saint when Scotland became an independent nation by the declaration of her growth in 1320. St. Andrews has a generous and cooperative spirit. He is the patron saint of fishermen, fishmongers, singers, and ladies, pregnant women, and is said to offer protection against sore throats and others. It wasn't until the 18th century that St. Andrew's Day began to become an annual cause for celebration. In 1792, a group of wealthy Scottish immigrants set up the St. Andrew's Society in Charleston, South Carolina, USA, to promote the philanthropic ideals of the saint. Further societies sprang up throughout the States and were famous for their protection of widows and orphans. In London, London Society, a members club near Hyde Park Corner holds a St. Andrew's Day every year. Our Rotary Club has surely had few Scots as members in the past, and many a Scottish speaker at its meetings. Amongst them was Sir Harry Lauder, the famous entertainer. That was in 1921. In 1926, when Stanley Leverton was president, John Reith was a member. He was an Aberdonian, the son of a minister of the Free Church of Scotland, and he later became Lord Reith, the founder of the BBC. Thus, Rotary has indeed, among many of its past members, a famous member, Lord Reith, indeed. Today, we have 4 Scots in our club: myself, John Buchanan, Tim Waugh, and Rosemary Bellow, who was born in Edinburgh. Of course, our senior honour member, Prince Philip, though not Scottish, is of course the Duke of Edinburgh. And let us not forget too that Paul Harris, the founder of Rotary, his wife Jean Thompson, she was a Scot, and her remains are interred in Edinburgh. And I learned this morning from Ian that The 30th of November is famous for another date, the birth of St. Winston Churchill. Thank you, Mr. President, for giving me that opportunity.
Thank you very much indeed, Past President Hugh. And amazingly, if our members look at last week's recording, they will find that you managed to do that last week as well. They could look at it and note one or two little differences, but thank you very much again for giving us that, talk in the flesh, as it were, today. I'd like to move on now and welcome all our visitors. And ask Clive, Sergeant at Arms Clive, if there are any visitors for the first time other than our speaker.
Yes, thank you, President David. So those of you who are visiting for the first time, can you unmute yourselves or just wave at me and I'll try and pick you out. I know the first one who joined us today was Vishnu from Malaysia. Vishnu, would you like to unmute yourself and just say hello?
Hello, hi everyone. My name is Vishnu Subramaniam from RC Subang, Malaysia. I am the International Service Director for my club. So thanks to Miss Nikki to giving me the opportunity to log into your meeting. So I like to see what the guest speaker going to speak. So great to see everyone in this Zoom, and thank you.
Thank you, Vishnu.
Hello.
Yeah, I'd like to say hello, John.
Yeah, hi, thanks for having me. I'm John Bevan. I'm Charlotte Bevan's brother, so I've come to watch her speak on prostate cancer. I know David as well, of course, from a very long time ago, so I'm a distant cousin. So good to meet you all. Thanks for having me on.
Good to see you, John. Thanks for joining. Mike Redding.
Hi, Clive. My name is Mike Redding. Hi, I'm Mike Redding. I'm living in Lyon, France. I'm an American, a transplant from New Mexico in the United States. And I'm pleased to meet you.
Good to see you, Mike. Thanks for joining us. Now, it seems to be everyone who's joining for the first time. We've got several people joining us again and coming back. That's rather nice to see you all. So welcome everybody who's visiting today. Thank you, President David. It's back to you.
Thank you very much indeed, Sergeant at Arms Clive. There's one visitor one member I'm looking for to come on the screen, and that's Margarita Grease Bison. So perhaps you let me know if she appears.
She's not here, but can I just say something else to David? You may not have picked it up. I'm sure we're all delighted to see John Buchanan back with us today.
Oh, and Mary as well. Yes, John, please let us see you in the flesh, as it were.
Okay. Now, John seems to have some difficulty getting his mic up, although we were talking earlier. Okay.
Thank you for that. In that case, let me move on to reports from the committee chairs. In particular, I will announce that we have Club Council meeting at 2:30, and hopefully all members of Council on this call can make that meeting. I'm aware of one that can't. And I'd ask now if Isabella, Chair of the Community and Vocational Committee, would like to tell us the latest position on the Christmas lunch.
Good afternoon, everyone. Thank you, President David. I have quite an update, so I'll just outline everything that would happen on the day. Firstly, the C&V Committee has planned the Rotary Club of London very first virtual interactive Christmas lunch, 14th of December at 12:30. We are pleased to inform that we have a splendid afternoon planned, an afternoon of music, intrigue, dramatics, magic, and a very impressive list of auction items. I promise you will be thoroughly entertained. Our entertainers are Alan Hudson, one of the top magicians in the United Kingdom. He's a member of the Inner Circle with a gold star rating. He has performed for A-list celebrities, Monday Night Magic in New York, appeared on ITV's Britain's Got Talent, ITV's Penn and Teller, Fool Us. Alan is an outstanding magician, comic, and mind reader. Be prepared to be amazed. Lizzie B, a professional stage actress, trained at the National Youth Theatre and National Youth Music Theatre, and the star of Hairspray at the London Coliseum, reopening April 2021. She has an angelic voice and her performance will be astounding. We have pianist Nilesh, a talented young musician whose highlights include performing with the Bollywood playback singer, touring with his band to Thailand and Europe, and working with the BBC. Ladies and gentlemen, be prepared to be transformed with the smooth and melodious sounds of Maestro Nilesh. The Salvation Army will entertain you with a newly released, newly recorded, and downloadable music from a plethora of local Salvation Army musicians. Notwithstanding COVID-19, they have endeavored to make this year the most wonderful time of the year through the wonderful sounds of music, even via downloadable means and Zoom. Ladies and gentlemen, as you are aware, COVID-19 has significantly affected everything we do, meeting friends, family, and even visiting loved ones in hospitals, hospices, and care homes. Charities' finances are drastically depleting due to the inability to meet and host face-to-face events that usually generate funds to assist various causes. For this very reason, we are hosting this year's Christmas lunch virtually, positively, with the hope to make a difference in the lives of children and their carers. Further, we are pleased to inform that all the funds raised from your registrations, additional donations, and the auction will be donated to children in hospitals and hospices and their carers. We cannot make the 14th of December a success and help those who desperately need our assistance and support without you. Together, we can be the change we want to see in our world. And I encourage you to support the Rotary Club of London's endeavor by donating to this worthy cause. Finally, for C&V committee members, our next meeting will be held January 2021. Date, time, and agenda will be provided accordingly. Thank you for your kind attention and have a great rest of the day. And please donate to this worthy cause. Thank you very much. Thanks, President, and thank everyone for your kind attention.
Thank you very much indeed, Isabella. Much appreciated. Now, are there other committee chairs who have a report to make? I normally give 2 seconds of silence to say thank you.
If I can say something here.
Rajiv, please do, yes.
We finally got to meet our 2 global scholars in the flesh. So Sabrina and myself took them out for lunch, a pub lunch in Hertfordshire. They were both doing well, and they both go home to their respective countries in the next couple of weeks, but they'll be back in January. And I've invited them to our Christmas lunch next Monday. Thank you.
Thank you very much indeed, Chair of the Foundation Committee. Are there any other comments?
Almoner's report.
Almoner's report, yes, please, Harry. Yeah.
Fine. First of all, good afternoon, everyone. I'm delighted to see John back together with Mary. I spoke to Nick Tash today, and he is comfortable. He is a prostate cancer sufferer, and he's been undertaking some treatment which makes him feel quite tired, but otherwise he's very cheery. Sadly, he lost the use of his legs because of back surgery. Nick and I seem to be going through a very similar set of circumstances. Anyway, he is okay. I've spoken to him. I've spoken to Flo Kaufman. She is okay. And finally, Vazi, unfortunately, had to go into hospital on Friday. She wasn't feeling at all well. She's at home again now. I think it's the long COVID which is affecting her. Some days she's good and others she's not. Yesterday she was feeling fine. Today she's feeling not at all well. And she sends her apologies for not coming today. So that's my report today, President.
Thank you very much indeed, Harry. It's always reassuring when you have little to say, but it's very good news about John, and a bit concerning about Vasi. Visits to other clubs, we've had a few. Does anybody who's visited another club on the internet wish to comment on that, or shall we move on? Birthdays. Our oldest voting member has a birthday this week. I would like to be advised which birthday it is. I don't know whether anybody knows, but Albert age, I know it's in the late 90s, but I'm not quite sure which one.
I think he's 97 or 98, David.
I think he's 97, which means he'll be 98 now.
Yeah.
Thank you for that. I have another conversation going on beside me, and that's why I'm just a bit distracted. Albert is our oldest voting member. He is not our oldest member, who is now in his 100th year, and that is HRH Prince Philip. We then move on to the next point, and that is to toast our first toast of the luncheon, which is to toast the Queen. The Queen. Those of you that have a vessel with which to do it. It now, I think, falls to me, Margarita, as one of our senior scientifically inclined members, was going to introduce Charlotte, and she's put some work into her preparation, but she has a problem connecting. So I will do the introduction. If Margarita comes online, perhaps she'll say a few words later. Professor Charlotte Bevan is not something I have called her to her face. Charlotte is a second cousin of mine, and I'm very, very pleased to be able to invite her to speak to us today. Her CV that you've had explains her 25 years of research background. What it does not explain is why I'm so pleased that she could join us today. Her father, my cousin, would be a little surprised if he was able to be with us still, but I think very pleased by Charlotte being invited to speak to us at a Rotary Club lunch. The fact that we do not meet in person and we do not eat in person would have been incomprehensible to her father. But as with her mother, he would have been immensely proud with what she has achieved and the manner in which she has achieved it. So I was going to say that at the end, and I'll have to work out what else to say at the beginning. But Charlotte is a senior researcher, 25 years solid research at Imperial College, dedicated her life to making the future for particularly the male gender, well, the life expectancy of the male gender longer than it presently is. And I really look forward to hearing Charlotte tell us about her research and what trouble she has had with testosterone. Charlotte.
Thank you, David. Right, I will just start. Right, so I'm based at the Hammersmith Hospital campus of Imperial College. I've been there for a little over 20 years now, imposing Victorian building. But David gave me a very broad brief to talk about, my career, my research, COVID, the problems of shutdown, etc. So it'll be a bit of a whistle-stop tour. I'm sure bits will be more interesting to some than others, and hopefully you'll find something that you can relate to. So I grew up in Cumbria. Rock refers to, actually, my career ambitions were initially to be a rock star, something I haven't quite given up on yet, but not happened to date. However, I was educated at the Cockermouth Comprehensive School, and actually sort of adopted by the largest employer in Cumbria, which is British, well, actually they are now, I'm not sure they were then, British Nuclear Fuels PLC, and became one of their management trainees. They sponsor a certain number of individuals from the area through university every year. So thus began my sort of start on nuclear physics. And that's what I began studying, King's College, Cambridge there. But that changed as I went through the natural sciences degree. I became more interested in the molecular biology side of things. And in fact, by my third year, I was specialising in genetics. Did a project then on Drosophila, these fruit flies in the middle here. Actually, if I just change this, hopefully you can see my pointer. So these guys. And interestingly, I was looking at a mutation which specifically affected male Drosophila, and thus perhaps began the sort of gender bias of my research career, as David has mentioned. From there, I went on to do a PhD in the School of Clinical Sciences based at Addenbrooke's Hospital. After that, I moved to postdoctoral research in London and moved from paediatrics, which is where I'd been based at Addenbrooke's, to cancer research. And that was initially at the then ICRF labs in Lincoln's Inn Fields. I don't know if you're familiar with those, the hideous lab building in that beautiful square, which has since been razed to the ground, and all the research groups have moved out to the Crick at St Pancras. And from there was recruited to Imperial, really to kickstart their prostate cancer research program, because there was very little happening at the time. So much so, in fact, that even though at the ICRF I was working on prostate cancer, I was doing that within a breast cancer lab, because there weren't many prostate cancer dedicated labs at all. And this is our current building. I've moved around a bit at Imperial, but we're now in a very beautiful new building, the Centre for Translational and Experimental Medicine. So my PhD, as I said, was in the Department of Paediatrics. It was on a condition called androgen insensitivity syndrome, which is a genetic condition whereby babies who are genetically male, they have XY chromosomes, grow up as more or less female in appearance. And actually there is a new face of androgen insensitivity syndrome, which is the model Hannah Gabi Odiel. She is an XY individual, believe it or not, so genetically male. And it's a very distressing but rare, fortunately rare condition. I moved into cancer because it's much more prevalent, and so felt it would have a greater impact. So I'm now working on prostate cancer. I'm going to talk a little bit about our work on COVID as well. So what on earth do all of these have in common, I hear you ask? And the answer is hormones. So hormones control a very great many different things, your moods, your development. Thanks. And the definition of a hormone is a signal that's produced in one part of the body that acts on other parts of the body, and they get there via circulating in the bloodstream. Now, the prostate is controlled by hormones, and the type of hormones that it's controlled by is androgens. Androgens are a class of steroid hormones. They're often called the male steroid hormones or the male sex hormones. Females have androgens too, just different amounts of them. And the androgen that you'll be most familiar with is testosterone. That's the major androgen in our bodies. And I have to be very careful when traveling. As I learned, the first time I ever went to the United States was for a conference in Dallas at the International Congress on Steroid Hormones. And being asked on immigration why I was there and giving the right response about steroid hormones, got a very funny look, and all my bags searched as the guy decided that steroid hormones were illegal, aren't they? So since then, I've been very careful to talk about cancer conferences rather than steroid hormones when traveling through customs. And I hope this isn't too simplistic for too many of you, but just to introduce cancer, I'm sure it really doesn't need much of an introduction. Cancer is really a group of diseases because it's a disease of cells, and all of our body is made up of different types of cells, and each cell type really has a different cancer type, unfortunately. It's a problem with cell division. Now, we need cell division. Cells, new cells are needed for us to grow, for us to heal wounds, and because cells have a finite lifespan, when they die, we need to replace them. But it's usually very tightly controlled. We have go signals, we have stop signals, and they are very, very tightly regulated. But they can go wrong in cancer. Maybe the stop signal has been lost. Maybe the go signal has been amplified, or maybe both. And this results in too much cell division and tumours forming. And the go signal in prostate cancer is androgens. So how do androgens work? How does testosterone drive prostate cancer growth? Here comes a little bit of science, sorry. So they work via a protein called the androgen receptor. And if we think of testosterone as a key, the androgen receptor is the lock. And it exists in cells, including in prostate cells. This is it here. Testosterone enters the cell from the blood and binds to the androgen receptor. Various things happen which end up with the androgen receptor binding to DNA. It's a class of proteins called transcription factors, and what that means is that they affect how genes are read. And what the androgen receptor does, with the help of other factors as well, is it promotes gene expression of genes which cause cell division in the prostate. And unfortunately, when there is a cancer there, they cause growth of the tumour as well. So it's unsurprising then that this is what we want to stop in prostate cancer. This is what a lot of therapies are aimed at doing. And current therapies act by removing circulating testosterone. That can be chemical, but also castration, the first sort of hormonal therapy for prostate cancer, very little used today, fortunately. Or a class of drugs called anti-androgens that bind straight to the androgen receptor and stop it working. And when I started my lab in 1999, we wanted to find new targets for therapy. And that's because although anti-androgens work really well, they only work for a given amount of time. And usually, I mean, it's good news in that men will survive long enough, but it also means that they'll develop resistance to these therapies. So we need new therapies as well. So we were looking at these cofactors that are needed for this to happen, and also at sort of downstream steps in the pathway for new targets. What happens if we switch off androgen action? Well, here's the prostate. It's at the base of the bladder surrounding the urethra. And that's the problem. If you get overgrowth of the prostate, it will sort of strangle the urethra and cause all of these problems with urination. Now, it's all very well switching off androgen receptor here, and the rest of the male reproductive tract even, but it's important in many, many other places in the body, including the brain, the bones, the muscles. It's not just in the men either. It's got male, female specific actions, and also obviously brain, muscle, etc. in the female also. The hair follicles even are responsive to androgens. Male pattern baldness is an androgen-dependent condition. So Charles Huggins won the Nobel Prize in 1966 for his work on hormone therapy for prostate cancer and breast cancer. And in his acceptance speech, he said, appropriate endocrine modification results in catastrophic effects on cancers of several kinds. The results are often spectacular. And that is true. The results are catastrophic and spectacular. So they, as I say, they work really well. But because of all these other tissues where androgen receptor signaling is important, there are a lot of side effects, and these can range from, you know, the embarrassing to the inconvenient to the life-changing and sometimes life-threatening. So very, very important side effects. And then to add insult to injury, as I say, these often stop working, resistance develops. So my lab and many others are really driving research into looking for new therapies. What do we want those therapies to do? Well, if we sort of take the lock and key analogy a bit further, a lot of these sort of blanket therapies that are used will affect all of these different aspects of androgen receptor signaling. What we would like is to find new therapies that are more selective, that maybe only target prostate cancer but leave libido and bone alone, or you might argue maybe target male pattern baldness also. So how do we go about it? Well, what we tend to do is to identify proteins or nucleic acids like DNAs and related molecules that drive prostate cancer cell growth in response to signals, mostly in my case androgen, investigate the mechanisms by which it does this. The more we know about how factor X does this, the more we can then go on to either target it as a therapy or use X itself as a therapy. And also, what we want more and more is to move towards personalised medicine, where there isn't a one-size-fits-all type of medicine, but we can look at a patient and say, okay, this will work better for you. And to do that, we need biomarkers. And there are lots of different biomarkers. There are diagnostic ones, which tell you whether you've got the disease, prognostic ones, which will tell you what the outlook is like, which we're very short of for prostate cancer, predictive biomarkers, which can tell what treatment might be right for a given patient, and biomarkers of disease progression that you can monitor to tell whether the treatment's working or not. Now at Imperial, obviously I have a prostate cancer lab, and we work really closely with the surgeons in urology, Hash and Matt, and the oncologists, of whom I don't seem to have any photos, apologies for that. But we don't exist even in that much of a vacuum. Imperial's a bit one institute, many centres. So I'll just point out some of these. We're part of 2 separate Cancer Research UK centres. One is Imperial specific, and another one is jointly with the Institute of Cancer Research, and they support a lot of research and give infrastructure. Another institute with the Institute of Cancer Research is the Movember Centre, which I jointly lead along with Johan de Bono, and it also involves UCL. We have a biomedical research centre and an experimental cancer medicine centre, which support studies and trials involving patients and provide a lot of support for those. And then we work closely with our colleagues. Breast cancer and prostate cancer have a lot in common, so there's a lot of synergy between research in those areas. So work closely with scientists in those areas. Imperial's got a real focus on multidisciplinary research, and so I personally collaborate a lot with colleagues in chemistry and in bioengineering and systems biology to develop new approaches to cancer research. And then obviously it's a global community now, more and more, and we work closely with colleagues in the rest of the United Kingdom and across the United States, Europe, Australia, and some of the most important collaborators are shown here. And I'll just briefly show you 3 vignettes from our ongoing studies. I've picked a few out to show you. So first of all, I want to talk about how non-cancer cells affect therapy outcome. This is work undertaken by research fellow Damien, who's supported by the Prostate Cancer Foundation. And cancer cells don't exist by themselves. Obviously, they're embedded in an organ, in a person. And this led to the seed and soil hypothesis, with cancer as the seed, but the sort of host cells, if you like, as the soil. And what Damien's interested in is, what does this soil do? How does it affect outcome? So if we think about the prostate itself, here are the cancer cells, these squiggly yellow things, but we've got normal epithelial cells, which are the cells from which cancer arises, and then normal muscle cells and other nerve cells, blood vessels, all in the same area and contributing in a way to the tumour. And then prostate cancer unfortunately metastasizes. It's almost unique in cancers in metastasizing to the bone, which is extremely painful. And it also goes to the soft tissues such as the liver and the lung. And all of these have their own different environment, different cells in there. And what Damien is focusing on really is the liver, because these visceral, as we call them, metastases in the liver and the lung really have been increasing in recent years, possibly because of the therapies, the long-term therapies that are being used. They could drive that change, possibly. And unfortunately, men with visceral metastases have worse survival than men with bone-only metastases. So we really want to understand what's happening in the liver and, of course, in the prostate. And is this due to changes in the microenvironment, soil, if you like? What are anti-androgen treatments doing to that? And how is it affecting the cancer progression? So men undergoing surgery, routine surgery, will kindly donate excess tissue to the lab. We can grow that in the lab, treat it with these therapies, and look at what changes happen. And Damien can do this by separating the different components, the cancer cells from the other cells, and doing gene sequencing to look at genetic changes. And also using some great new imaging equipment, which we just got to look at the protein changes. And this is a whole slice of prostate where we can see different proteins sort of in situ, if you like. And this is going to give us a greater understanding of how the disease itself is evolving in response to the treatments that we're giving it, and also help us to really allow clinicians to be more informed about what treatment is going to work best for which patient. Next, I want to talk quickly about obesity, which is a really important risk factor for prostate and other cancers. Now, 1 in 6 men in the UK will be diagnosed with prostate cancer in their lifetime. They are not necessarily going to die of it, because prostate cancer is sometimes a very lazy disease. It's very slow growing, and you'll die with it rather than from it. And we call these pussycats. But there's another form of prostate cancer, call them tigers. They're more aggressive, they will become lethal. And currently, when a man's diagnosed, we've really got no way of telling whether he's got a tiger or a pussycat. But obese men are more likely to get the tigers. They're not more likely to get prostate cancer, but they're more likely to die from it. So we want to understand why that disease is more likely to be lethal. And this is a ticking time bomb, because currently 1 in 3 men in the UK are obese, and it's rising. So you can imagine the intersection, the Venn diagram here. We've got a large intersection, and obesity really has become identified as a major risk factor. So if you look at this, here's a prostate at the base of the bladder, as I showed you before. Here's a tumour. There is fat surrounding the prostate, as all our organs, and our hypothesis is that this fat is releasing factors which can communicate with the cancer cells and encourage tumour growth. And the types of factors are sort of mini cells, if you like, which contains genetic material. So we're looking into what factors are released by the fat cells, which of them are taken up into the cancer cells, and what does it do to that cancer cell growth? And do these factors differ between the fat that is next to the prostate and fat that's further away, and between lean and obese men, to try and give us an understanding of what's going on here? And this is undertaken by a urologist, so a surgeon in training, who's undertaking Ph.D. He will consent men who are given their diagnosis and agree to us using excess tissue from their prostatectomy, so when their prostate is removed. They'll donate blood, but also prostate tissue and fat tissue, which is rushed to our labs. It has to be because the surgeries take place at the Charing Cross Hospital, which is in Hammersmith, and our labs are at the Hammersmith Hospital in White City, rather confusingly. So that is rushed to us, and we then grow that in the lab, and we can extract from this the factors that are released by the fat cells. This is an electron microscopy image, hot off the press, of these mini cells. And as well as all this high-tech equipment, I just want to say Joe has been really proactive in making his own equipment. So he's devised this cell, to isolate these factors, a little sort of prototype using Lego here. He made this in the Imperial Innovation Rooms at White City, which were part of this new campus that we've opened up. And they're fantastic. You can go in and build anything, use equipment there, and it's open not just to staff, but to members of the public. So I really recommend going along if you're in the area. You can book in there. So what this study is going to show us is, well, first of all, we're establishing a unique biobank of matched samples, thanks to the generosity of the patients, which will be a unique resource for this study and other studies. We're increasing the education and the research in this area. This is one of the very few studies looking at obesity and cancer, and could even give us evidence to lobby government to do more about the obesity epidemic. And identifying these factors is going to allow us to identify new therapies for prostate cancer, which will benefit not only obese men, but also lean men. And finally, our labs were closed completely from the end of March to the beginning of July. We were only allowed to go into the labs for essential COVID research. Well, you can imagine that was a challenge to us. And in fact, we did find some use for our research in COVID. So this is another work in progress. So the COVID virus, SARS-CoV-2, enters your lung cells using your own proteins, which is a terrible shame. So the two lung cell proteins that it uses are TMPRSS2 and ACE2. They exist on the cell surface. TMPRSS2 actually modifies the virus. It primes certain proteins, these spiky things that you see in all the pictures of COVID, and allows them to interact with another host cell protein, ACE2, which then takes it into the cell. So therapies could involve inhibiting TMPRSS2 or ACE2, but the inhibitors that are available for these proteins are not great. They have bad side effects, or they're really not very good drugs. But we looked at this, and TMPRSS2 is very familiar to us. We look at it in prostate all the time because it's an androgen-regulated protein. If we give androgens, TMPRSS2 levels go up in prostate. If we give anti-androgens, they go down. So can we use this in COVID? Nobody knew what it did in lung particularly, but our hypothesis was that if we give anti-androgens, these drugs used in prostate cancer treatment, will get rid of TMPRSS2 in the lung cells, and the virus won't be able to be primed, so it won't be able to go into the cell. So before we could even go into the lab, we could do a bit of computational work with existing data. Labs share data all the time. So looking at somebody else's data, we could see that the androgen receptor and TMPRSS2 and the other one, ACE2, are all expressed in the same lung cell types, and they're all expressed in the types of cells which are targeted by COVID. So that was a good proof of principle. And the models that we can use in the lab, mostly we use cell lines. So we took a lung cell line and we can see androgen receptor in there. Great, we knew we could then do research on these cell lines, and we've expanded to other cell line models since then. Now, if we look here on this graph, the higher this bar is, the more TMPRSS2 there is in those cells. If we treat these lung cells with an androgen, DHT, it goes up. And this was the first sort of real study of this. But then if we add in an anti-androgen, one called enzalutamide, one called bicalutamide, it goes down. So for the first time, we'd shown that TMPRSS2 levels in the lung can be controlled by anti-androgens. And this is also showing it in some lung tissue. But what does it do to viral entry? Well, initially we used a sort of dead version of the virus, a pseudovirus, and we showed, so here this bar shows you how much virus is going into the cells, 100% in the case of no treatment, but down to less than 50% when we gave these cells anti-androgens, bicalutamide and enzalutamide. We'd steered clear of using live virus, as you'll understand, but actually a collaboration with Wendy Barkley in the Department of Infectious Diseases, she's on the SAGE committee, so been very busy with all the COVID research, allowed us to use live virus, and hot off the press we can show that this live virus cell entry is also halved by anti-androgen treatment, which is fantastic. So we're hoping that this will be published very soon, so that it will provide evidence for trials into use of anti-androgens in COVID. What we think is that using these in sort of early hospitalized cases will reduce the severity of the infection. And why is it good? Well, because these drugs are already licensed for use, so there's less clinical trial necessary. And also they're produced in large volumes because drug companies are using them in hundreds of thousands of men every day. So that could be scaled up for COVID use. And further, in short term, they're pretty well tolerated. They don't have such bad side effects on sort of short-term treatments. Now, all our research, David wanted me to talk a bit about funding, and we're funded by charities in the UK and in the US, the research councils, sort of small foundations, private charities, and also pharmaceutical companies over the years, which is fantastic. And we do our bit for these as well. We're involved in outreach. So for Prostate Cancer UK, we've made videos. For many of these, we've had lab tours. We raise money for Movember. I've gone and addressed the March for Men that Prostate Cancer UK have every year. We've had some celebrities around the lab. Claire's very keen on this picture of her with Peter Andre, for example, who's a great ambassador for Cancer Research UK. Of course, charities are in major trouble, as you have already pointed out today. And so it's more and more important that we sort of help them in their fundraising efforts, and we're very engaged with that. My lab's grown over the years. I couldn't find any really early pictures, but if we look at it growing from 2011, to the present day, some of the faces sort of stay the same, but mostly people stay for 2-3 years and then move on. And this is the latest picture of the lab. We're really dependent on scientists from all over the world, and long may that last. Obviously, we're hoping that it does. And it's great to work with such an engaged team of young scientists day in, day out, really, really inspiring. But of course, as well as my research, I've got other roles in the university. And I was just sort of jotting down a few of them here. So there are roles in teaching, which is, again, fantastically rewarding, in training, in governance, very important, a lot of admin roles in the various institutions and centres that we're involved in. Some of you will recognise this. It's an award which is given for gender and diversity sort of issues. So we have to prove that we are an equal opportunities employer in order to get this badge. They go bronze, silver and gold. And It's a lot of work actually to apply for these and to be awarded them. We're very proud of our Silver Swan status. The government's just announced that it is removing the necessity to have this status in order to get certain grants, which we think actually is a bit of a retrograde step, and we're not going to let it prevent us working towards our gold award. So all of these things, juggling all of these roles is very challenging. And part of it is that the Athena Swan mandates that we need to have equal representation, of course, on all of these boards and committees. Now, there aren't that many female professors, and so those of us that there are probably do more than our fair share of this type of work, sort of the collegiate part of our jobs. And the challenges of COVID, well, as I say, it's fantastic working with this team of people, and we have vibrant lab meetings every week and a lot of seminars. Sometimes we get treats like pizza at our lab meetings. So this is a lab meeting last year, and this is what they look like now. So in a way, it's easy to gather people on Zoom. Certainly we found that for the senior academics, we get better attendance at meetings than we used to. For my lab, it's a shame that we can't meet more in person. At the moment, we're only allowed 30% of the capacity of scientists in the lab. And so even if we were all to try and meet up in person, it would only be 30% of us at any one time. So we're not going to stop Zoom meetings anytime soon. Conferences is another big part of our job. Traveling to these meetings, it's not just a jolly, it's an important way to meet and network with scientists from all over the world. I was really fortunate. I organised this meeting, which happened at the end of February, so we just got it in under the wire before travel was restricted, and it had participants from all over the world. It was held in the Bahamas, and this is, you know, another part, not just listening to the talks, but meeting people. So this was in the bar after one of the talks. I'm meeting up with Ava from Spain and Ian from Scotland, and on the back of that, we're currently writing a paper and a grant application together. These are the types of things. And also you get to meet, you know, last year we met Jim Allison, just as he'd won his Nobel Prize for immune checkpoint inhibitor research. This meeting 2 years before this meeting this year looked very different online. It was still a great meeting, but really not the same. You can't meet new people and new collaborators. Teaching, weirdly, has adapted itself quite well, I think. The course that I'm mostly involved with has made it its business to be have a new model of teaching, less of the didactic lecturing and more interactive, so that the students are researching themselves, feeding back to each other, teaching each other. And so this is one of our classes last year. They're in groups, and then we put them into teams. We have little quizzes, we have competitions, and then, you know, they'll go back and research and then feed back to each other. This is a group presenting what they found. Now, that works quite well online, but I've got to say, for the teachers, it's perhaps less rewarding because, you know, students aren't very keen to switch their cameras on, and very often it feels like you are lecturing to a brick wall. It's hard to get back that feedback, get that real sort of buzz going in the room, but we are certainly trying our best. So on that note, I'll leave it. Thank you very much for your attention. Happy to answer questions on any of those aspects. Sorry, I've probably gone on a little too long. As I say, there was a lot to fit in, but I hope some of it was interesting to at least some of you. And I will stop screen sharing.
Thank you very much indeed, Charlotte. Certainly, I won't say what I felt about your talk at the moment. Let me see whether there are any questions. And let me ask Clive if he would act as a question master for us.
Yes, I will do that, President David. Please, if you wish to ask a question, either raise your hand on the reactions button and a little blue hand will come up and I know that you want to ask a question. Did you want to ask a question, Tony?
Yes, please.
Yes, please go.
Professor, that was a fascinating talk. When I was fortunate enough to get a knighthood, my children immediately renamed me Circumference Hippo. So I think we can be confident that I am that 1/3 of the male population that is clinically obese. But not all clinically obese men get prostate cancer. And I was just wondering whether at any time do you need men over 70 to volunteer as samples so that you can work out what it is that might trigger in some men who are obese to be more prone to cancer than others?
Well, absolutely. For certain studies, that would be fantastic. We're not doing, we're sort of not powered at the moment to do the sorts of studies that you might want to volunteer for, such as gathering data on you. What we're doing actually involves gathering fat tissue. And I don't imagine you would want to volunteer for that unless you were undergoing surgery for something else.
Right.
But what we're hoping is that what our research will give the sort of evidence needed to do one of those larger studies and to get the sort of statisticians and epidemiologists involved. And in that case, then absolutely, yes, indeed. Great, thank you.
Well, I have Hugh and I see Anthony Newton, so I will do Hugh first, please, and then Anthony Newton. Hugh.
Fine, thank you, Charlotte. For several years, I have a 6-monthly routine test for cancer. And of course, the usual PSA test as well. And so the last time I saw my consultant, he said to me that if a man lives long enough, inevitably he will get prostate cancer.
Yes.
Would you like to expand on that?
Yes. So the prostate is dependent on testosterone for growth during development. So in the womb, the prostate develops and it stops growing soon after you're born. It grows again at puberty with a huge hit of hormones that happens then, and then it stops growing. It should then not grow in response to testosterone. But when a man hits about his 40s, it almost inevitably starts again. So we get a sort of second wave of androgen-dependent growth. And if you were to look in the prostates of men in their 40s and 50s, about half of them would have some sort of growth of their prostate that shouldn't be there. And if you were to look in the prostates of men in their 80s, about 80% of them would. So it sort of keeps going up. So he's right, it would get to 100% eventually. Not all of that is cancerous growth. Sometimes it's not malignant, it's benign. But even the cancers, as I've said, they can be these pussycats which won't ever cause any sort of clinical problems. The problem is once it's diagnosed as cancer, we really don't have at the moment, unfortunately, much way of telling between them. But yeah, absolutely. Prostate cancer is a huge problem. It's just, in fact, overtaken breast cancer as the sort of biggest killer of, so it kills more men now than breast cancer kills women. It's second only to lung cancer and then cancer death.
Thank you. Anthony Newton, and then be followed by Jim War. Anthony, your question, please.
Thank you very much, Charlotte. Wondering whether Brexit will inhibit the collaboration between countries at all, and also to what extent, if at all, you work with China?
Right. Interestingly, my first trip to China was it was last October. So just before all of this broke out. Working across the world, obviously, it's Europe that will be threatened by Brexit, the rest of the world hopefully not affected. Most of my PhD students at the moment come from Europe. I fear that we will be restricted to a much smaller talent pool, if you like, because the fees that overseas students pay are at least 3 times as much as home students. And EU students are going to be liable for those overseas fees from June. So we're not really expecting to get many applications from EU applicants for PhD places. Now, hopefully the UK population will step up, but it's unfortunate. We'll have to find other ways of interacting with the rest of Europe because we really don't want to lose those connections. And as I say, that talent, you know, it's been really beneficial to the science to have those European students and postdocs in the labs. China, I was sort of just starting to talk to them after my trip there. Obviously, that's been slightly put on hold. Imperial has got a lot of links with China. There are specific studentships which are only open to Chinese nationals. So we'll see how that progresses, I think. When I was visiting China, they were very keen to start collaborating more with the UK. I think it was because they were very annoyed with the US at the time.
Okay. And Russia, what about Russia?
I actually don't know many collaborations with Russia. actually, personally. I'm sure there are some, but not within my division at the moment.
Thank you. Thank you for that. Tim Waugh, and then Carl Roberts, and then Harry Simmons, and then Brian.
Thank you very much for an interesting presentation. My father had problems with his prostate. I had a prostate operation a few years ago, which was the most wonderful thing that ever happened to me. I have two sons in their 40s. And I mean, it seems to me there's a genetic element to this. What do I tell them at this stage? What should they be doing?
There definitely is a genetic element. We don't really know which genes are involved. I mean, there are genes that are linked with prostate cancer, but there are so many of them. It's not like you could have one genetic test that will really give you the best answer, unlike in breast cancer where you can have the BRCA test. Although the BRCA mutations do predispose to prostate cancer. So what your sons should probably be doing, and I have talked to my oncology colleagues about this, is that they should have a PSA test probably now in their 40s, certainly by 50. Not because it's expecting to detect anything, but to give it a baseline so that then they can go back and get tested maybe every couple of years or so that can be kept an eye on and you can get an early warning if anything did develop.
I had heard that PSA wasn't very effective, and there are other ways of testing. Is PSA still the most effective way of doing this?
Yes, you're right. There are other tests. PSA itself isn't so much used anymore. They do things like PSA velocity. It's the same test, but they'll do different sort of calculations from it. So they can be a bit more sophisticated about the PSA test. There are other genetic tests that you can get, not really yet on the NHS, you can pay for, or there are trials going on at the Institute of Cancer Research led by Rosalind Eales. I think it's called, there are so many of them. There's the Profile trial, the Protect trial. She does studies into family history, and anybody with a family history is eligible to enter those trials. So that you could, if that was something that was of interest, that's something to look into. Yes, you're right, though. We're always looking for better markers than PSA, more specific ones.
Thank you very much.
Thank you. Carl Roberts, then Harry Simmons, then Brian Summers. Carl, if you'd be so kind to put your question. Carl?
There's a belief that many cancers are increasingly triggered by what we eat today. Is this fact or fiction? And compare this with the fall or rise of testosterone levels in men.
Yes, that is a very interesting question. So there are a lot of sort of papers come out linking certain foods to risk of breast cancer, prostate cancer, etc. Not many of them have been studies done in a big enough number of people to say for sure. But things that have been linked to prostate cancer are a high consumption of meat and dairy products. And it's said that a vegetarian diet is protective. So a vegetarian diet will cause you to have less risk of prostate cancer. The one thing that's been definitively shown to increase your risk of prostate cancer is a diet high in saturated animal fats, dairy fats. And it's certainly shown that the incidence of prostate cancer in Asian countries is much lower, but that if people from Japan have moved to the United States, their risk of prostate cancer goes up, probably because their diet has changed and other environmental factors as well. Now, you said to try and link that to testosterone. Well, if we think about obesity, for example, hormones are made in the fat tissue as well as in the testes, etc. So it's possible that you could get higher hormone levels. That's certainly an issue in breast cancer when after the menopause, the fat is the major source of estrogens in women, for example. Now, I don't think we know for sure yet whether there's something similar happening in prostate cancer, but that's a possibility.
Thank you.
As a cancer survivor myself, I'm quite pleased to see the correlation and to be reassured that your research is pointing us in the right direction.
Great, thank you. And the positive thing is that even in people who have cancer and who are cancer survivors, changing the diets can have positive effects and reduce risk of recurrence and improve response to therapy. So there's always something positive to be done.
Thank you.
Thank you. Harry Simmons, and then Brian Summers.
Thank you very much. Do you have a website for lay people like myself who are also prostate cancer sufferers, so we can keep up to date with what you're doing in fairly lay terms?
Right, good question. My lab itself, we don't, and I think that's something that we should think about doing, and so I will sort of try and instigate that. Thank you. The best place to look for a good lay summary of a lot of the research, not just mine, but stuff that they fund and across the UK, is Prostate Cancer UK. They have very good sections on ongoing research and also information for prostate cancer sufferers and helplines as well. They're a fantastic charity with a lot of information.
Thank you. And also, there are 3 vaccinations or immunizations for COVID coming out at the moment. Is there one which is particularly more beneficial for prostate cancer people like myself?
Interesting. I've been wondering that because obviously certain vaccines can't be had by people with compromised immune systems. Now, the ones that are coming out at the moment, I don't think that there's any reason why you wouldn't be able to have them, but I'm sure that that will be one of the first things, because obviously people with underlying health conditions will be high up the list for these, so they'll be careful filtering of which vaccine is best for people with different conditions. To my knowledge, I don't really know of any reason why these ones that are the Pfizer one should not be fine to have, but I'm sure that's something that they're looking at very carefully.
Thank you.
Thank you. Brian, would you like to put your question, please?
Thank you very much. Professor Charlotte, that talk was absolutely amazing, one of the best I've ever heard, and thank you for that.
Thank you very much.
My question is, throughout your talk, you put the emphasis on weight and not on smoking. Now, virtually the entire country and almost the entire world thinks if they don't smoke, it's a deterrent to getting cancer. But you've emphasized that weight is for this type of prostate cancer is the most important. And I think that's something that needs to be promulgated and emphasized, because I don't know anybody that thinks weight affects prostate cancer. And so we really need to look at this in a different way, I believe.
Yeah, I think you're right. Cancer Research UK did try and have a bit of a publicity campaign when these first publications came out about obesity being important for not just prostate cancer, many different cancers. I don't think the message has got across yet. You're right, smoking is becoming less of a threat as smoking declines, but obviously weight is going up, obesity is becoming more of a problem. And so that was one of the reasons that we're keen to get our obesity research, out there and properly funded and talked about, because as I say, this could be information to lobby the government to try and educate people about the risks of obesity. It's not just to do with heart disease, huge problem there, but also cancer.
Thank you very much indeed.
Thank you.
Thank you. You wanted to come back, Brian?
Thank you very much, Clive. Professor, one of the things I've always reassured myself with is that you think if you get prostate cancer, yes, it may kill you or it may not, but it's a slow process. However, sometimes I read in the newspapers or on television, some poor chap got prostate cancer about 6 weeks ago and he's just died. Now, of course, that takes away that, shall we say, that kind of level of slight security. Can you comment on that?
Well, I would say presumably they were diagnosed with quite advanced disease, which still happens. So when I first started 20 years ago or 25 years ago in prostate cancer research, I think about 60% of diagnosis were of men in whom it had already spread and metastasized. It's much less than that now. People are generally caught much earlier. But obviously men still may, because it's an asymptomatic cancer often, there are no external signs, you may not be diagnosed so early, so it may already have spread to the liver, to the bones. So they'd be very unfortunate, but that could be what happened in those cases.
Thank you very much.
Thank you.
I'm not necessarily saying to go off and get tested because, as I say, the PSA test isn't particularly specific. It might tell you that there's a problem with your prostate, but it won't tell you whether it's cancer or not. You can't tell that unless you get a biopsy done, and that is not the most pleasant process. You know, it's an operation to remove some of the tissue, and it's got its own problems of infection, and so it's something that really needs to be discussed with GPs, and I wouldn't advocate rushing off to do it sort of on the basis of this, but something needs to be carefully thought about. And like all testing, you need to think about, well, what will I do with that information? And these days, actually, the imaging for prostate cancer, I've not spoken about it because I don't do imaging research, but I mentioned Eric Ebawaji, who does a lot of PET imaging for prostate cancer. I showed one of their images in the corner there. Now, before getting biopsied, getting an imaging, a PET or an MRI image will help to non-invasively give an idea of whether there's cancer there or not. And actually, we're lucky in London. There are a lot of centres that can do this. The rest of the UK may not be so lucky. So that's definitely something to consider.
Thank you. Well, unfortunately, time has beaten us. And President David, can I hand back to you, please, for your thanks and final comments?
Thank you very much indeed, Clive. And indeed, thank you very much, Professor Charlotte. I will ask my Vice President to give the vote of thanks for your talk, but I just wanted to give you my personal thanks for coming and speaking us today, and to reiterate the pride I'm sure, and I'm thinking here of your father particularly, would have in us having this conversation now, and in having being aware of your presentation. So thank you for that. Vice President Ian, would you give the vote of thanks, please?
Yes, President David. We've already said the pride that you and your family take in Professor Charlotte, and we're very lucky that you have that connection because we benefited from hearing her speak today. I know, President David, that you gave a wide-ranging brief when you asked her to come and speak to us, and Professor Charlotte, you've met that wonderfully today, and we've all benefited as a result. You were not only able to explain to us the research in respect to prostate cancer, the research that you've carried out, but also the funding issues and also management, which you also get involved in. And the slides that you gave us an enormous amount of information in clear and precise manner, and probably answered a lot of questions that perhaps people have either been afraid to ask or look into before. And so you brought that over in an excellent way. And then you're able to bring us right up to date with COVID-19, not only the research that's being carried out and the results from that, which I think are ahead of the press, as it were, and we may well see that in the press soon, but also how your research and the studies have been affected by that. You showed pictures of the group that you established in 1999. And it just shows how successful you have been in expanding that group and attracting researchers from all over the world. So I know, Professor Charlotte, that President David and all the members of the club wish you every success in your future research and in everything that you do, because we have been very impressed by what we've heard today, and I'm sure that members will join in with me in thanking you accordingly.
Well, thank you so much. Thank you, David. Thank you, Ian. Thank you, Clive, Margarita, who I spoke to yesterday, and all of you for your attention. It's been an absolute pleasure. I'm sorry I didn't get to meet you in person over lunch, but who knows, maybe in years to come. Thank you very much indeed.
Thank you, Charlotte. Now I have the final task of your presentation, of presenting you with a certificate which confirms that we are thankful for your presentation, which I will be sending to you digitally and perhaps physically shortly. it now leaves me just to thank you and to move on to the closing of the meeting. To thank also the family that appeared, but my brother Martin, your brother John, and to thank you for that. So let me move on to the next item on the my agenda, and I'd like to firstly invite Tim McAndrews. Tim.
I'm here, David. Yep.
Would you give us some insight to why we're all so excited about next Monday's lunch?
I certainly will, President David. I think everybody is aware of this, but of course the Rotary Club London Christmas lunch is one of the centerpieces of our club each year. And of course for David, who has had a pretty tough time running this club on Zoom, it is his first major social event. So I will implore you to join us. There is a joining fee, as you will have seen. We're charging the CMB committee suggesting a charge of £35 to join, which is what you would normally pay to come along to a Christmas lunch. It's just this year you don't actually get the lunch included. But Isabella and her team in CNB have gone to an enormous amount of effort to pull together a magician, the Salvation Army band, other musicians, and in terms of the auction, and I will be the auctioneer next week, I think she's got over 20 items now. So there'll be a lot happening. Please do try to join and come along. And I look forward to seeing you all then. Thank you, President David.
Thank you, immediate past President Tim. And so it moves on to my announcing that the next meeting, of course, is our Christmas lunch. But the next speaker in January will be our own member, Doug Wills, who is the managing editor of the Evening Standard, talking about the issues, if you like, of print publication at the moment. So thank you for that.
David, normally we advise people what the auction prizes are, and I think all members should be aware of this before the auctions, if that can be arranged. I think that would be very beneficial in collecting money.
Brian, there is a Candy Liu is producing an online auction list that I'm sure will be circulated. I'm hoping that people who can't make the auction will put in a bid that cannot be exceeded in person. So I'm hoping for a competitive auction and raising as much money as we can for our charities. Bearing in mind that even your contribution of £35 registration for two people, if there are two people there, is a contribution to charity. But we will be doing our best with that. Thank you. So, to the final toast then, Brian, if I may. To Rotary and peace the world over.
Rotary and peace the world over.
Now, and the bell. I close the meeting.